Hello grade 12s and welcome to Life Science, and today we're going to have a look at revision of meiosis. Now before I start, remember these two words that sound similar. You get meiosis and you get mitosis. Okay, now remember mitosis, the one with a T in it, always tells you two identical daughter cells. Okay, meiosis is the creation of 6 cells. Okay, so don't forget it, meiosis. So if we have a look at the board, meiosis, there's no T involved. Okay, the nice thing, I always remember it by the T meaning two identical. Okay, so please don't forget it. Now, let's have a look at it. When I have a look at revision, okay, especially with a section, the best way to have a look at it is going through questions over and over again. Okay, so this whole section is just questions. Okay, so make sure you got your thinking caps on and let's see if you can get them all right. Okay, first one. Let's have a look at this whole piece. How do I know? Well, let's just first look at the question. The diagram below shows 2 daughter cells formed at the end of meiosis 1. Okay, the first thing we need to understand is how do we know meiosis has taken place? Okay, now I'm just leaving that because that could be a question later, so I'm not going to answer it yet. All I want to know is, can you see meiosis has taken place? Look at it very carefully. There's a reason for it. Okay, now I'm going to leave it. Let's have a look. The question is, name 2 errors that lead to unequal number of chromosomes in cell A and B. Okay, so you need to name the error. What has happened? Why has it done this? Okay, and I'm going to give you a minute to do that. So give me everything you got, think about it. It's a simple word. Are you ready? Off you go. Okay, let's have a look at it. Now, same picture, there's questions there. All I've done is I've had a look at that. Please look at this very carefully. Okay, there is one of those. Okay, have a look at all of them. There's one that looks all different. Can you see they're all different sizes, thicknesses, everything? The problem we have here is there's 2 of those. Can you see it? And look at this part here. Okay, can you see it? That tells you something has gone wrong, especially when they said it's meiosis 1. It means something has already happened. Okay, this whole process that's happened here is called non-disjunction of chromosomes. In other words, the 2 chromosomes haven't split apart to create to go into 2 separate cells. Okay, in this case, you're going to get a disease. Okay, it depends on which chromosome or which chromosome number or pair of chromosomes that this actually happens. Okay, so it's pointless me telling you what it is because we don't know which chromosome. It could have happened on the 24th pair. Okay, oh, 24th pair, how can I think? The 23rd third pair. It could happen on the 21st pair. It could happen on the 1st pair. Okay, so there's lots of ones that we've got to have a look at all times. But it's not a problem. Let's just carry on. Still the same picture. Okay, we've got a couple of questions as we go on the same picture. Here we go. During which phase of meiosis 1 did the error mentioned in question 1 occur? Okay, and explain it. Okay, there's no explanation there. I just need to know the answer. Always give the answer, and then I want you to try and tell me, why do you think it happened there? How do you know it's happened there? Okay, so I'm going to give you a minute. Are you ready? Off you go. let's have a look at it. Now, what part of meiosis does this happen? Okay, this is called anaphase 1. Can you remember how to remember all the phases? Okay, now it's normally known as IPMAP. Okay, so let's just put here IPMAT. Okay, now when it comes to meiosis, this part is where the problem comes in. We don't actually have that. Okay, and normally it's, if you look at mitosis, it's interphase, prophase, metaphase, anaphase, telophase. In this case, you get prophase, metaphase, anaphase, and telophase. Okay, this whole thing, this anaphase, which is this part here, okay, the anaphase over there, what happened was when they were lined up on the equator, right, and you have the spindle fibers all coming down, as they were pulling apart, okay, because they were lined up with homologous pairs, can you remember? And as they get pulled apart, instead of one going that side and one coming this side, both of them pull down here because there was disjunction on there. Right, so we have a little bit of a problem there. Okay, so this happens on anaphase 1. Please remember, there's no interphase in meiosis. Okay, now, carrying on with the questions. Now, you got to understand that I can ask millions of questions on a specific picture, which they are going to do. Let's have a look at this one. What was the number of chromosomes in the parent cell before the start of cell division? Okay, how many chromosomes did the parent cell have before this division took place? Okay, make sure that you give me one answer, make sure you know exactly what it is, and let's see if you can tell me why. I'm going to give you 1 minute. Are you ready? Off you go. Okay, let's have a look at this. Now, if I have a look at the two, remember, meiosis, you're going to start with one cell over there, and it's going to divide, because remember, this is meiosis 1. And you're going to end off with 2 cells over here. Okay, now the question is, how many chromosomes is in this parent cell before the end of meiosis? Okay, before the end of meiosis 1. So this is very, very simple. If we count here, 1, 2, 3, 4, 5, which is a problem. 1, 2, 3, 4, 5, 6, 7. There's an extra chromosome on this side, which means supposed to have one over there, and this one is supposed to disappear. Right. That means it's 6 and 6. Now what happens in meiosis 2 is, remember, we're halving the number of chromosomes. Okay, so let's have a look. It's going to be 12. The parent started off with 12 chromosomes before this process took place. Okay, one of the big things you must remember on what happens at each stage. Okay, now, still sticking with the same picture, if we have a look at this, what would the chromosome number at the end of meiosis 2 in cell A? What would be the number? Look at it carefully. What is the number of chromosomes at the end of meiosis 2 in cell A? Now, I'm going to give you 1 minute, but remember, you need to tell me why. I'll give you the answer and we'll talk about why, but you need to tell me why. Are you ready? Off you go. Okay, let's have a look at it. Now, remember the first part of meiosis 1, certain things happen and something happens at the second one. How many is here at the moment? There is 5. Remember, meiosis 1 and meiosis 2, the difference is one is the division or the minimizing or making half the amount of chromosomes, and the other one just keeps the same. So at the end of meiosis 2, the number of chromosomes is going to stay as a number 1. Okay, so let's have a look. 5, because there's 1, 2, 3, 4, 5. Remember, we already know this is at the end of meiosis 2. Okay, remember when it comes to this, you're just going to get random assortment. They're going to line up on the equator randomly and get pulled up to their certain sides. Simple, easy, no fussing. Okay, so you carry on thinking about it this way, everything's going to happen quite easily. You need to know a lot of different informations about these things to be able to get to where you want to go at the end of the section. Okay, here's another question for you. These are, by the way, these are questions that come out of past papers. So you know they're going to be asking things like this. Here we go. What would have been the chromosome number in each of the daughter cells if the error mentioned in question 1 had not occurred? Okay, what would have been the chromosome number in each of the daughter cells if the end of at the end of that you mentioned in question 1 if question 1's problem did not occur, if non-disjunction did not occur, if everything was perfect, what would the chromosome number be in both daughter cells? I'm going to give you 1 minute and explain your answer. Are you ready? Off you go. Okay, now if I have a look at this, the simple question that came through, okay, you need to understand you're not babies anymore, right? So if I have a look at this, wait, let's just bring there, what would have been the chromosome number in each of the daughter cells if the error mentioned in question 1 had not occurred, okay? This is very simple. These are the 2 cells, right? Remember, we're going to create 4 cells at the end, every one different, okay? In this case, you will notice that here, 1, 2, 3, 4, 5. This side, 1, 2, 3, 4, 5. That would have made it even, and there's 2 more. 1, 2. So that means there's 7. Here is 5. How do we make them even? They've got to be even. They've got to be 6 each, right? So this one should have been 6. This one should have been 6. Therefore, the chromosome number at the end should have been 6. Okay, very important to remember at the end of these sections all the time, they're always working in pairs roundabout. Okay, so I know that doesn't sound right because it's not, but you've got one cell. It divides equally, even though the DNA is not the same, okay, or the material has changed. If there's 10 up here, there'll be 5, 5. Okay, it's not going to be 10 up here and there's going to be 4, 5. Can you understand what I'm trying to get? It's got to be even. There will always be exactly the same number of chromosomes in each one of the daughter cells. Okay, never not the same. Okay, so after going through all of those questions, I think you need a break because I definitely do. So I'll see you straight after the break. Cheers. Welcome back. Hope you had a good little break, right? And you got all your stuff sorted. And if you had mistakes in the last one, you knew how to fix it. You wrote down all the information that you actually needed for each and every one of these slides. Now, excuse me, I've got another slide. Have a look at the slide. It's a quick summary of things, okay? It says study the diagram below, which shows a process occurring in human males, right? In a human male. Now, I'm going to bring that down to about there. Please understand, this has got a certain process. This arrow represents, I always call it the PMAT. Remember, it's prophase, metaphase, anaphase, telophase, okay? That's what happens at this arrow here. Then you're going to end off with a cell. Then the cell's going to happen again. You're going to get your PMAT. Okay, let me bring it all there. That's PMAT, creating your prophase, metaphase, anaphase, and telophase, and then creating 4 cells that are unidentical. Okay, because remember, this is a certain process. Now, how's this for a question? Simple. Name the process by which male gametes in a human formed through meiosis. So what is the process's name of making a male gamete? Okay, I'm going to give you 1 minute. Give me the process. Are you ready? Off you go. Okay, let's have a look at this. You guys are not babies anymore, right? You are grade 12s, which means you do not use little baby words anymore. So if I have a look at this whole process, the process that we go through when we go through meiosis was only in males, not with females, in males, right? It's not called making sperm. It's not called making gametes. Okay, yes, it is making gametes, but it's not called making gametes. Okay, and it's not just called sperm. Okay, you've got sperm involved. That's perfectly fine, but you need the rest of it. Remember? So it's making of spermatogenesis. It's called spermatogenesis, is making sperm cells. Okay, spermatogenesis, genesis. Okay, spermatogenesis is the one that actually makes a sperm. And of course, which one do you have? Spermatogenesis. So what would be the females? Think about it carefully. Okay, remember, yes, they make eggs, but they're called ooh. Okay, so they're called oogenesis. Oogenesis is the making of an egg or the female gamete. Makes sense? Okay, all of this is very important to remember names. Remember, in life science, we always use biological terms, and you need to know that. You can't use the baby terms anymore. Okay, now, let's have a look at these questions. Same picture. You see meiosis division 1, meiosis division 2, and there's A and B. And then it says there, name the organ in the male where this process mentioned in question 1 takes place. Come on, that's easy. I'm going to give you a minute. Are you ready? Off you go. where does sperm get made in the male? Okay, now remember we have done, you know, the male reproductive systems. Okay, so you've got your epididymis. Remember, it gets made, matured there, and stored there. But okay, so it's not there. It gets made inside the testes. Okay, remember, inside the testes. And can you remember what actually makes them? You've got special glands. Remember, you get your vas efferents, which joins it from the testes to the epididymis. You've got your, what are those, spermatogonia cells? Those are the ones that make them. Can you remember? You've got to remember that all sections come together. So in this stage, they can say to you, which cells make sperm cells? And you've got to go spermatogonia cells. You need to be able to remember exactly which one does where and how it goes. And lastly, testes means two, okay? If we're talking about one specific one, it's called a testis, okay? It's an IS, so it's testis, okay? Which means one testicle, okay? As long as you remember that, very important to remember. Can you remember what makes the females? Which goes through oogenesis. It is your ovaries. Okay. Next slide. Still the same one. Question. How many chromosomes will you find in each cell at A and B? So I'm going to do that. I'm going to go A and B. How many chromosomes will you find at A and B? How clever are you? Are you ready? I'm going to give you a minute. Are you ready? Off you go. Okay, now let's have a look at this. How many chromosomes will you find in each cell at A and at B? Now, the problem is, you need to understand, you're going to look at me and go, are you mad? Look at that. It's just a circle. I don't know how many chromosomes are in there. There could be 2, there could be 100. What's going on? You need to read the question very carefully. If we go up here, human male. Okay, you need to read questions carefully. So they try and trick you like that. So that means if we bring it down, in number A will be 46, okay, and B will be 23, half the amount of chromosomes at the end. Okay, don't get confused. Don't lose it. The question was there, how many in a human cell? Okay, it doesn't matter if it's male or female, they both have the same, right? But we need to have a look, 46 in the one, 23 in the other. Okay, you guys getting it? It's very easy, it's very simple. I know you guys are working on it and it's becoming very easy for you. Now, here we go. Same thing. Next question. Name 2 processes occurring during the first meiotic division that contributes to genetic variation of cell A. Okay, hold on. What I want you to do is I want you to give me that and an explanation about how those 2 work. So what I'm going to do is I'm going to give you the process name and then we're going to talk about it. So what I'm going to do is I'm going to give you, let's give you 2 minutes, okay, a minute for each. I want to know the what that is and how does it happen? What does it do? Okay, so I'm going to give you 2 minutes. Are you ready? Off you go. Okay, let's have a look at this. Now, very, very simple. The two processes that occur during the first meiotic division that contribute to genetic variation of cell A. So we're just looking at this one. Okay, there are two. First one will be crossing over, and second one is random assortment of the chromosomes. Now, what is crossing over? Remember, I gave you 2 minutes on each. Okay, crossing over is where you get the homologous pairs standing next to each other. Right there they are. And I'm going to take a red one here. Come on. And this one. Oh, wait, that wasn't red. It wasn't very clever. Let's go red here. So I'm going to just get rid of that. We're going to have a red one. And yes, they're homologous pairs, which means they have the same set of chromosomes. Okay, let's lengthen this page a bit. No, that's not what I want. No, let's go back. Let's lengthen this page so we can actually see what we're doing. Okay, you got your homologous chromosomes. You're going to get your blue ones again. There it is. This one's going to go there. And if you look at this, I'm just going to bring that one out. Let's leg out a little bit to make your life easier. The red one is going to do the same. It's going to come down and out there a little bit. Okay, so that is crossing over. And at the end, you're going to end off with something that looks like this. Okay, and the blue one will look, first the green, yellow one there. I'm just going to join those in there for the fun of it. That one there, and then this one there, and the red. Can you see how they've changed to the original ones? Okay, that's how they do crossing over. Because of that, it's got a little bit, there's a little bit of red on the blue one, and there's a little bit of blue on the red one. That is called swapping genetic information. It's called crossing over, and the main part where they cross over, that little piece over there, so if I color it in nice and red there, that piece there is called chiasma. The place where crossing over takes place is called chiasma. Okay, that is the first one. And the second one, what happens is random assortment. Okay, random assortment means they line up in any specific way. It doesn't matter. Okay, so some of them will have the full color up here and then the half color. So I'm going to put like that. And the others will have the half color up there and then the full color down at the bottom. Okay, so they line up randomly so that when they move to the different poles, everything is different. No matter how they try, if they line up differently or line up in randomly assortments, anyone goes anywhere and it gets pulled up to the side, and then that child is special for that specific DNA. Right, does this all make sense to you guys? Okay, those are the two things that make meiosis so special. And you must understand that there is lots of different characteristics on each chromosome. So don't just think that one chromosome is going to give a difference, okay? A little piece, like, you know what, all I can say is a millimeter, but the millimeter, it's much smaller. A small little piece of it can change things like your eye color, hair color, and skin color on a specific little locus, on a specific gene, right? It becomes so delicate and so amazing. Right, so we finished that. I'm going to give you a quick break because I definitely need one, and I'll see you straight afterwards. Cheers. Welcome back. Now we're going to carry on with the same type of questions with the same type of pictures. Okay, so here we go. Let's have a look what I have here. How many cells at B will carry the Y chromosome? How many cells of B will carry the Y chromosome? I'm not explaining anything more, okay? What I want you to do is I want you to tell me, and I want you to be able to explain to me how is that possible, okay? And therefore, I'm going to give you 2 minutes, okay? In actual fact, I'm going to give you 3 minutes, okay? 3 minutes because I want 1 minute to tell me how many Ys, and I want you to quickly explain to me how is that possible, okay? Excuse me, 3 minutes. Are you ready? Off you go. Okay, so let's have a look at this. I was a little bit naughty when I came to this question, especially now that I've been asking you, because I thought that we'd bring in another section while we're busy doing this. Okay, so the question here was, how many cells will B carry a Y chromosome? What does that mean? First of all, let me give you the correct answer. It's 2. Okay, and second of all, let me explain why. Remember, boys have XY chromosomes and girls have XX. Remember that? Okay, remember we did this thing called genetics, right? And in genetics, if you take an XX, which is a female, okay, I'm going to put the F, and I'm going to put XY, which is a male, yes. Remember when we go through meiosis, remember that part? I made sure that you remember it's got to go through meiosis, and we're going to make gametes, which is an X and an X for the female, and the Y and the male will be X and a Y. Okay, then they're going to join together, and you got this thing called a Punnett square. Can you remember your Punnett square? This is a very simple one. This you can just do this. Okay, this will be X. Versus a female. Male will be X, Y, and we just fill it out. XX, XX, this one will be XY, XY, and if you have a good look how many we've made? We've made 4. We've made XX, we've made 2 of them, and we've got XY, and we've got 2 of them. So, let's just pretend, okay, you have XX, XY, XX, XY. Okay, that's if this was joined, okay? In actual fact, you're going to end off with something that looks like this. Okay, because remember, there they are. Well, that's the females, and that's the males. We're looking at males because we're looking at spermatogenesis. Nice and simple, hey? Don't forget it. You need to remember that with DNA. They can always ask you the process of meiosis during your genetic sections. Okay, very important to remember that, and don't forget it. Now, next question. What are mature cells at B called? What are the mature cells at B called? You've got 1 minute. Off you go. Okay, this one was very easy, okay? And if we have a look at it, what is the mature cells called? They're called sperm cells, spermatozoa, or the male gamete. Any one of the 3. Those are quite simple and easy. Okay, now, here we get to the more complex little things. Have a look at that diagram. You need to know what is happening there. Okay, and if we have a look at it, it says, identify the phase represented by diagram B. I'm giving you a minute, and you need to tell me why. Are you ready? Off you go. Okay, let's have a look at the answers of this one. If it'll move, okay, remember, this is metaphase 1. How do we know it's metaphase 1? Okay, mainly because we said diagram B, that's how we know. Remember, there they're lined up at the equator with their homologous pairs. Can you see it? Crossing over has just gone, and it's crossing over has just happened, okay, and have a look how it's lying up, okay. If it was lying up with that piece there being white or dark and every other piece being white, then it would be random, or it wouldn't be random. Can you see how it's random assortment? Because it's lined up in opposite ways. Can you see that? Very important. Okay, let's go to the next one. Question is, explain why homologous chromosomes in diagram B have mixed for genetic material for each chromosomes. Okay, are you ready? Off you go. 1 minute, sorry, by the way. 1 minute. Are you ready? Off you go. Okay, let's have a look what I have here. Okay, oh, come on, let's move. There we go. Okay, very simple. Crossing over has taken place, that's how you know, and genetic material has been exchanged. Remember, we want to make things different, that's why we're exchanging, and random assortment is happening. Right, next question. I'm going through these quickly now, because we haven't got much time, so I want to see how many you can get. Name the phase that will follow the one represented at diagram A. I just want the name of the phase. 1 minute. Are you ready? Off you go. Okay, this is very simple. The one that's going to repeat diagram A will be your anaphase 2. Very, very important. Anaphase 2, you need to represent the 2. If you don't represent the 2, we're in trouble. Okay, make sure you always put down which phase, 1 or 2. Next question, here we go. Describe the event that takes place in phase mentioned in the previous question. Okay, describe the event that takes place in the phase in the previous question. 1 minute. Are you ready? Off you go. Okay, so let's have a look at this. The question was, name one, no, sorry, wrong one. Name one place, no, back, there we go. Sorry, I've got mad. It says here, describe the event that takes place in the phase mentioned in the previous question. This is quite simple, okay? And I'm going to go through this as nice as possible. You've got the spindle fibers contract. Okay, remember the spindle fibers are those fibers there that are joined. The centromere splits and pulls the daughter chromosomes or chromatids to opposite poles of the cell. So as those spindle fibers start shortening or contract, they pull all the different chromatids across, okay? They get put over there, and then that huge, magnificent thing called cytokinesis begins, okay? Remember, there's two. Cytokinesis is the splitting of the cytoplasm, and that's when those two cells, because all of this happens in one cell, and as it gets pulled, all the chromosomes get pulled to the one side, this starts going in like this, which is called cytokinesis. What is the other one? Can you remember? What is the splitting of the nucleus? It's called karyokinesis. Well, I hope I've helped you out and you guys remember all of this stuff, okay? Remember, study hard, and remember the basics. Always the basics come first. Okay, until next time, cheers.